Hello, everybody. This is chapter 5. We're going to go through the different sorts of eukaryotes of microbiology and just talk about their general qualities. So the way this chapter is organized is by the kingdoms, Protista, Animalia, and Fungi. In the Protista, this is a very mixed bag of organisms because they don't really fit into classic fungi, animals, or plants. So they resemble characteristics of each, but they are not true organisms in those different kingdoms. So we have the Protista kingdom, the Protozoa subkingdom, which is animal-like. And they're animal-like because they are heterotrophs. So they need to eat other things in order to survive. So hetero is other and troph is to eat. So they have to eat others. They are unicellular, and most of them have some sort of motility, so they can move around. In algae, these are plant-like, so they are photosynthetic, and they are unicellular and multicellular, because if you think about seaweed's an algae, and it's really large, so it's multicellular. The small ones that we can't see with the human eye, in the ocean, they tend to float on the top, so we call that plankton. A lot of plankton are in this subkingdom. And the term zooplankton refers to those little organisms that are like animals. So they basically are heterotrophs and they need to eat other things. And then phytoplankton, the phyto refers to photosynthesis, and so they are able to photosynthesize. The subkingdom slime mold are fungal-like. So fungi are heterotrophs. Fungi always have to degrade and degrade the living things that they're on, or the things that they're on, sorry. So you can find molds, anywhere, and they always destroy the things that they're growing on because they have to break them down and feed. Slime molds are the same way. There's two kinds of slime molds in the world. There's an amoeboid, which is like an amoeba, and then there's another form called a plasmodial form, which is very interesting. They form colonies, and they look like, I guess they look like in the forest floor, you would think that they look like lichen, perhaps. We'll look at some pictures of those. The kingdom Animalia, the one that's going to be talked about is our helminths. And then kingdom fungi, they're yeasts, and there's some molds that can infect humans. So we can start off with just talking about what kind of hosts, because parasites are always going to enter into a host, whether that host is a cell or the host is a whole organism. So the term definitive host refers to the one that the parasite produces sexually in, male and female. Gametes are made there. Just gametes are made there, excuse me. Gametes are made. The intermediate host is something that the organisms will go into and infect, but they reproduce asexually. And some parasites are very complicated and have more than one intermediate host. And then you have your accidental hosts that we sometimes are that victim of. And it's not part of the true parasite's life cycle, but accidentally we can ingest or take in a parasite, and then they grow inside of our bodies as well. So you can see this is an example of Plasmodium that causes malaria. And just to point out that there's a mosquito and there's a human host. And we can see that in the mosquito, we are creating male and female gametes, right? So this would be then the definitive host is the mosquito. And then when they infect a human, they reproduce asexually. So it's an asexual reproductive style. No need to memorize the life cycle. Just know that this is an example of definitive versus intermediate host. So for life stages, we call the living, feeding, growing form of a protozoan a trophozoite. So it's kind of the same meaning of a vegetative cell in bacteria, because it's metabolizing, it's alive, it's growing. And then the cyst, some protozoans can form cysts, and it's very similar to an endospore of bacteria because it's a dormant stage. So a cyst is a cell, it has a protective wall to protect them from the environment, because again, just like endospores of bacteria, the trophozoites will form a cyst when the environment becomes uninhabitable, right? There's something that is not good for growth, and so they can form a cyst, and we call forming that cyst encystment. And then when conditions come favorable again, they excyst, so it's excystment. This is just a nice picture of Entamoeba histolytica, which we will be looking at later in the semester, the cyst form and the trophozoite form. So it's kind of similar, but yeah, one is a cyst and one's the trophozoite. So we'll learn the differences later. Okay, so looking at the protozoa, there's many ways that we can acquire protozoal infections. Sometimes they can go through mucous membranes like the eye. Sometimes they are vector-borne. Vector means that there is usually an insect that helps to transmit the disease. So we have, for one, right, we'll just keep with the mosquito that transmits the plasmodium that causes malaria. You can see there's other ones too. You can actually have some that can upon contact, they can actually penetrate the skin. Some of them have, enzymes that can degrade the skin to get in. We will study and look at schistosoma in the future. Sexual contact, ways, again, mucous membrane, right? So Entamoeba again, or not again, but Entamoeba, Giardia, and then there is a sexually transmitted infection called Trich for short, but Trichomonas is the organism that can cause that. Fecal oral, you can see there's a lot in this category. Fecal oral means that the infectious either egg or cyst or the trophozoite itself is ingested. It's lost through the feces. So that an organism, an animal, or a person that's infected would have some form of it in their feces. And then through unsanitary conditions, someone else could pick them up in food, contaminated water, and ingest it orally. So fecal oral transmission means that the pathogen comes out through the feces and then ingested orally by accident. And then some of them you can inhale and then get into your lungs. So many ways that these guys can get in. Protozoans are so various that they have grouped them into like modes of locomotion. These guys can move around by cilia, right? So cilia, again, are small hair-like extensions. There's usually very many cilia. This is an example of a paramecium. And so it helps them move around. Pseudopods are a kind of extension of the cell membrane that helps them crawl around. Amoeba is a big category that move through this pseudopod method. And then we have, of course, the flagellum that helps to propel our cells. All right, another interesting thing about protozoans is that some of them have the ability to not reproduce just like one at a time, but reproduce in a very a burst of many, many cells. So this is called schizogony. And if we look at this is again an example of malaria, where we have the mosquito stage, right? And then we have the human stage down here. And I just want to point out the word schizont, right? So when cells are infected, the parasite will just basically make many, many, many cells, divide multiple times, and then be held within this one unit. And so the unit's called the schizont, and schizogeny is the process of making all of those different cells, or all of those multiple times of division. This is an example of a ciliate, right? So this is our paramecium and some specialized organelles that they have that you should know. Some of them have a primitive mouth called an oral groove. So they are heterotrophs, right? All these are heterotrophs, so they have to eat other things. And then they also have an anal pore to excrete the wastes that they have. And then one interesting thing they have is a contractile vacuole. And these contractile vacuoles are present because these are freshwater organisms, and through osmosis, the inside of the paramecium here, for example, is packed full of proteins and different minerals and ions, and so it's very dense with solutes. The water, by comparison, is not. So water is going to flow into their cells just by osmosis. And so with this constant flow of water into their cells, they actually squeeze it out. So they have these specialized organelles that fill with water, and then they squeeze them and they just push out the water. There's a video of this. Oh, let me show you the video. It's a fast video, but these two dark circles, you can see how they are squeezing. So that is their contractile vacuoles pushing out the water that is coming into their cells through osmosis. So that's an interesting organelle that they. All right, so our first example is amoeba. Amoebas are, in the environment. There are many of them, but there are a few that are virulent enough to cause disease. So that word virulent means the ability to cause disease. And so the more virulent something is, the more capable they are at causing disease. So very few are virulent enough to cause disease, but they do cause a lot of diseases. And a lot of people carry around some kind of amoeba in their guts, in their intestinal tract. The transmission, again, is fecal oral, or you can ingest the cyst, like we saw this guy, Entamoeba histolytica, can make a cyst in the other slide. In our flagellates, we have this example of a euglena. And one thing that you want to know, another organelle, is called a stigma, which is here. And it's actually a little eye spot. So these guys, single-celled organisms, but they have the ability to detect light. So these guys are interesting because they also photosynthesize, but they're still classified under protozoans because they also are heterotrophs. So these guys do both. So these guys will eat and photosynthesize. So that's the reason why I have this, because they need to look for light. Another thing that's mentioned here is the pellicle. So a pellicle, protozoans don't have a cell wall, but they do have a structure and rigidity to their cells. They have a shape, and that is due to the pellicle. So pellicle is underneath the cell membrane, and it helps to give the shape for the cell. You can kind of see the pellicle, see some bands here. You can see some of that like light green shimmery band underneath, just like this line here, that would be the pellicle. And for the group apicomplexans are really interesting. They're called this because the api refers to the apical complex, and apical means the top, just the apical surface of a cell. It is the top of the cell. So they have this complex that exists on the top of their cells, and they use this to infect the host. So apicomplexans always have this apical end that helps them infect. And this again, this is Plasmodium. Plasmodium is the one that causes malaria. All right, now we're moving on to slime molds. So slime molds are the fungal-like group, and these are the two different kinds of slime molds. So we're not going to spend a lot of time here because they don't really cause a disease, but there is a cellular sort of slime mold, as you can see here in letter A. It looks like a cell with lots of extensions. And they are basically individual amoeboid cells. They're amoeba-like as well. And they stream together, and they can sort of coordinate, sort of like forming a larger superstructure or superorganism, and they communicate with other cells. So these guys are really interesting. And so they have this really primitive multicellular aggregation that consists of individual cells that they have their own nucleus, but they work together. The other form of slime mold is called a plasmodial slime mold, and these guys are large. Brightly colored organisms consist of a large cell with many nuclei, and these are the ones that are really visible on the forest floor. So when you look at this, if you see a slime mold, you don't really know what you're looking at, but that is a nice image of a slime mold. Okay, so we're moving on to our parasitic helminths. These are worms. And for life cycles of helminths, so worms have an egg, a larva, and an adult form. Some of them have separate sexes, male and female, or they can have both in the same worm. And if they have both, they're called a hermaphrodite. They have a lot of different kinds of life cycles. It depends on the worm. They usually have more than one intermediate host. And the biggest source of a worm infection would be untreated human feces. So fecal oral transmission. And, anywhere in the world where the water system is not tightly regulated, you can have contamination, or if you're out in the field, in the nature, you can drink contaminated water. And so how you get a worm infection, right, is you accidentally take in something very, very small that you can't see. So you're going to ingest by accident the eggs or the larva. Some of them can actually puncture through the skin, but mostly we'll be talking about the ingesting the eggs of the larva. So once the worms get into your body, they do grow really big. Like tapeworms can get 6 feet long. So how do they stay in your body, right, with little detection? So a lot of them have a specialized mouth. We'll look at the tapeworm because most worms are going to be intestinal. Some of them do travel to other organs, but we're always, moving. There's blood coursing. So there's a lot of activity and movement in the body. So they will have a specialized mouth just to attach to the intestinal wall. So if you're thinking about the intestines, your intestines are always undergoing peristalsis, squeezing the waste and food through our body. So if the worms were not attached, they would just be lost with our waste. But they do attach and they stay present in the body. Some of them will have produce enzymes that can liquefy and penetrate the tissues. So sometimes when they get into our body, they can travel this way, or they can get in through the skin this way. Some of them have a cuticle, or this covering that basically masks or cloaks them. It just makes them undetectable by your immune system. So your immune system doesn't recognize anything on the cuticle that's foreign. So your immune system doesn't even know that they're there. And it's pretty remarkable that there's very few symptoms produced by these helminth infections because they have all these different ways to evade the immune system, to keep quiet, because they are really good at what they do. They're parasites. And so if a parasite were to make a person very, very sick, that's not a very good parasite because the person would either die, right? And then the host would be gone, or have, clues to their infection and alert the immune system, which could take care of it. So very few symptoms with these guys. The two groups of helminths that you want to know are nematodes and platyhelminths, otherwise known as roundworms and flatworms. So nematodes are unsegmented. So think about a pencil, just like a cylinder. And it does have a full digestive system, meaning it has a mouth on one end and it has an anus on the other end. Even when they're parasites, they do that. They're very common in terms of intestinal parasites. And what usually happens is their eggs, they live their life inside your intestines and they produce eggs, and eggs can be identified either in the feces or sometimes around the anus. An example of this, you may have heard of pinworm, perhaps. Pinworm is a very small roundworm that does get detected. Their eggs actually get detected around the anus. Ascaris lumbricoides is something you probably not have heard of, but we'll look at that later. Trichinella is the source of a pork infection from eating raw pork. And then of course there's some worms that like heartworms for dogs. So worms can infect a whole series of different kinds of animals, not just humans. Platyhelminths are flatworms. There are two kinds of flatworms we're going to talk about, flukes and tapeworms. Flukes are also known as trematodes. Tapeworms are also known as cestodes. And yes, you need to know both terms. Flukes are non-segmented. Again, they're just one sort of oval-shaped flatworm. The term that's flat is like, yes, they are literally just like a flat organism. And they do have an oral sucker to stay inside of organs. Sometimes they have two. And then there are all different kinds of flukes. So flukes can actually, they can stay in the intestines. They can go to your lung. There's a lung fluke. There are blood flukes. There are liver flukes. So they do travel through the body sometimes and they infect organs. Tapeworms or cestodes are segmented. So this is our first and only segmented flatworm. And they have suckers and hooks, or hooks, but the scolex is a term you need to know. The scolex is the head region of this tapeworm, and it has those attachment suckers or hooks to attach to your intestines. And then the rest of the tapeworm is just filled with these segments, right? Because we have a segmented flatworm. Segments are called proglottids, and they're basically just male and female sex organs, and they just produce eggs. So the proglottids are just this unit that just continuously makes eggs. So here are some pictures. This is the pinworm we talked about. So you can see the inch here, the size comparison. They are very small, but they are a cylinder, so it's non-segmented. This is just a close-up of a pinworm. The pinworm name is Enterobius vermicularis, but we do not need to memorize that for this test. We will talk about this later. And this is an example of that pork trichinella, where there is this example of a cyst. So some some helminths, when they enter your body, they can encyst in some tissues. So the way that this life cycle works is that you put, and again, you don't have to memorize a life cycle. I just want an example of a cyst. So one you might, so let's just do this. Start at number 1, eggs and egg-filled proglottids. So these are just little tiny segments of the worm are passed into the environment. And a pig would pick up this, the eggs, right, from eating food in a contaminated region. And then once the tapeworm gets into pork, it forms a larva. Those eggs hatch into a larva. The larva then will penetrate into the wall, and so penetrate the intestinal wall and migrate to other tissues. So this would have those enzymes that can dissolve the wall. And what happens to these pork tapeworm in particular, it encysts into muscle. So this is sort of a dormant structure that you find in skeletal muscle. And as we do, we eat the muscle of pig, right? And when we ingest this in raw or undercooked contaminated meat, then we can get the tapeworm. And it does the same thing where it is ingested in our intestines, and then it comes out into the larva form, moves its way through our body to our skeletal muscle, and then it can encyst there. So, we're an accidental host. The pig is actually the host here. And this is a example of those Cysterici visible in infected meat. Okay, so one thing to mention here is if you've got pork like this, you would not want to eat it probably, but also like if you do cook the pork at the correct temperature, it does kill this the larva here. And this is an example of our tapeworm morphology. So again, our tapeworm is segmented, so you can see how we are picturing the tapeworm, and each segment, right, is called a proglottid. And proglottids have both testes and ovaries, so they can fertilize, like self-fertilize, right? So this is a hermaphrodite, and it's just full of eggs, right? So this is a gravid. All that means is that it's full with eggs. And so each segment of a tapeworm is just doing the same thing. So it's basically an egg-making organism with a tiny scolex, a tiny head. And remember, the scolex here has oral suckers and hooks. So this is a close-up of those features. So these are the hooks, and this is of a tapeworm, right? We see the word scolex. And then we have also suckers to hold on to our intestinal wall. This is a fluke. We can see we also have an oral sucker here. And this is another image of flukes, just to see their overall shape. They have sort of this like oval shape to them, and they're not very large. You can see about 3 centimeters, about an inch long. All right. Some flukes mate for life. So a male and female. Some of them they have different sexes. And so this picture is of a larger male fluke, this male, and then we have this groove that fits the female. So we have a little bit of the female that we can see here, and they just stay like that, and they just make eggs, constantly make eggs. And we can also see a really nice image of those oral suckers there. All right, then our third group here is our fungi. So fungi have two sort of forms to them. So fungi have a cell shape that is a little bit rectangular, and that is called a hypha, okay, hyphae or hypha. And the cells will sort of build themselves into these long threads. And so this is under the category of molds. Some of our hypha do not have those walls in between each cell, but we do see all the different nuclei, right? So there's many nuclei, but there's no wall between them. And so we just call that non-septate, right? So we have septate hypha and non-septate hypha in our molds. In a different fungus, right? So when we look at fungus, it's molds and then yeast. Yeast are different. Yeast are usually a unicellular organism. So, yeast is just a little circle. But when it grows, it can form pseudohypha. So it kind of looks like it's forming these threads like these two in the molds, but they're not true hypha. But they can take on that pseudohypha look. So fungi are chemoheterotrophs. Okay, so chemo refers to the fact that they have to eat chemicals to get energy. And chemo refers to the energy, meaning that they get their energy through breaking down chemicals. Okay, so like we do, we are also chemoheterotrophs. So we need to get our energy from breaking down the food that we eat. And then the heterotroph refers to the fact that where do we get our organic molecules? So this is going to be strictly organic, right? So organic molecules like sugar, for example, like glucose, we cannot just make glucose from inorganic starting products. We have to take in our organic molecules from the food we eat. So this is the same way that fungi metabolize. And so mostly our fungi are saprobes. This is a term that means decomposer. Okay, so they decompose dead things. Saprobes, so I'm just going to saprobes decompose something that's already dead. All right, they are going to digest something that's dead. So you can think about finding like mold or mushrooms on sticks in the forest, or bread, that's already, non-living. Some of them, right, some of our fungi are parasitic, meaning that they are going to feed on living hosts. So that's the big difference. Saprobes are going to live basically by decomposing dead things already, and then parasites have to infect something that's living. Yeast, so molds are aerobic, right? So we know that means that they need oxygen in order to survive for their metabolism. And yeast are facultative anaerobes. What this term means is that they are able to do both use oxygen when oxygen is present, and when an oxygen is not present, right, they can also survive by something called fermentation. So we'll look at fermentation in this unit. But facultative anaerobes, they love to use oxygen when it's present, but if you seal off a facultative anaerobe like yeast in an anaerobic environment, like a sealed jar, they can go ahead and ferment and still be alive. So yeast are going to be used to ferment foods, because fermentation is a process that, doesn't use oxygen. So beer and wine, for example, yeast are used to make beer and wine, and those things are made in sealed containers, right? Because we want to force them to ferment. Fungi also have evolved to occupy little niches that are not going to overlap that much with bacteria. So Fungi usually grow better at a lower pH, which is usually too acidic for bacteria. They are pretty resistant to osmotic pressure. So osmotic pressure means that the water is going to be drawn out of your cells through osmosis. But it doesn't really bother the fungi. So they are able to grow in high sugar or high salt concentrations. For example, jam, right? So if you have some jam at home, jam is basically fruit with a lot of sugar, right? So if we have a really high sugar content, most bacteria, that's the osmotic pressure is going to be too much for them and it dries them out, right? We looked at plasmolysis as a way, what happens to bacterial cells when they are put into a hypertonic environment. So plasmolysis will pull the water out of bacteria and kill them. So making a jam or jelly or canning any kind of, well, no, canning's different, but using those solvents to preserve food like jam is literally just using this high sugar content to kill the bacteria. However, it does mold. So if you leave some jam in the refrigerator long enough, right, you can find that it might get moldy because fungi are able to grow in that really high osmotic pressure environment. Fungi can also grow in like pretty low moisture areas, generally too low to support bacteria. And then they're also able to metabolize really complex carbohydrates. And for example, I have lignin, which is wood. Okay. It is the carbohydrate that, bark is made of. And if you think about after like a hurricane, people's houses have flooded. The biggest concern is mold because once you get moisture into the wall, right, that can be completely moldy. Like mold can grow on any surface seemingly and digest that surface. So you can get moldy books, moldy walls, wood of the house. It's just these guys are really very good at living and decomposing objects or substrates that bacteria can't. So why do you think these traits evolved, right? So it seems like there's a lot of comparison to bacteria because both of these organisms, molds and bacteria, are microbes, right? And they're going to have a competition between them to grow in the environment and grow on substances. So it's basically a way that fungi have evolved to use other means of nutrition and to grow in places that you don't find bacteria. So it's a way that they don't have to compete with bacteria. They do, obviously, but it's also something that gives them a leg up into growing in things that generally bacteria can't grow in. The morphology of fungi, again, when we look at a mushroom, for example, you can see this picture of a mushroom and how we have this string-like, spaghetti-like feature. This is the hypha, right? So the hypha, those little square cells, they form really long fibers, and that's what creates the mold or the mushroom. So the part of the hypha that grows inside the substrate. So this, if we think about like a log in the forest, sometimes we see this part, right? We call this a mushroom. That is a fruiting body, and that is basically just the reproductive structure. So this is mainly for reproduction, and they reproduce by making spores, and the spores would be all along the gills of the mushroom. So this is literally just to reproduce. The other part of the organism that's much, much larger than a mushroom is underneath the substrate that they're growing on. So if we're talking about a log in the woods, right, this would be underneath in the wood. So it would be basically driving its way into the wood, decomposing and degrading with enzymes, the lignin and other carbohydrates that are there, and feeding. So this network of hypha inside of the dead thing, right, the wood, is called mycelium. So it's not a root. It's kind of feels like it should be a root, but it's not, right? It's not a plant. So it's called mycelium. And the fruiting body, or the thallus, is used to make spores. And again, we see this picture of two different hypha, septate hypha and the non-septate hypha. That is how we can classify some of our mushrooms and our molds. That's it. So, okay, we already looked at that. Sorry. So this is a picture of the, different morphologies. This is a mold growing on a Petri plate. So you can see the fuzzy kind of texture that it has. It's fuzzy because it's kind of like carpet. If you look closely at a mold, you'll see that it has a long stalk, and then it has this round, like almost like a lollipop. So the stalk grows up, and the round part here is going to make spores. So spores for mold is how they reproduce. Mold and the mushrooms that we have out in the wild, they all produce by making spores. In this picture, this is our spore making part of the mold. There are different terms for this. We'll learn that later, but this is what's going to make the spores, and the spores are super tiny, really small. And the reason why they are lifted up and produced at the very top is so that when wind blows, you walk by, that those spores are going to just, be liberated by the draft and by the wind. How do they reproduce? They can do both asexual and sexual reproduction. So remember the two terms. Asexual means you're making a clone. So the genetics, if you're reproducing asexually, you will have a clone. You'll have exactly the same genome in the daughter cells as you do in the parent cell. Sexual means that you are, basically the DNA is getting mixed up with some other DNA from a different cell. So the DNA is going to get recombined. You're going to gain a different genetic profile than the parent, and that's what we call sexual reproduction. And, right, our spores are so lightweight that most of our spores, most of fungi depend on like the movement of air, wind to disperse them in the environment. So they're very lightweight. They're widely dispersed in the environment. They're in the air. They're in the water. They're on living things. So spores are everywhere we look. Fungal spores are very distinct. They have their own shapes. And so mycologists will use those spore shapes to classify them. Myco, just to highlight this, MYC is the prefix you use for fungi. So a mycologist is someone who studies fungi. So we want to remember MYC is referring to fungi. So all of this is talking about molds, right? Molds, or our fleshy fungus, which is our mushrooms, for an example. There's all kinds of fleshy fungus. There's like bracket mushrooms and puffballs. But they all reproduce by making these structures that produce spores. Yeast, however, is different. Yeast is a single cell. So yeast is called a unicellular fungus. This is what B1 yeast, and yeast don't produce spores, they bud. And what budding means is that you're going to have an asymmetrical reproduction. So you'll start to have a smaller cell develop on the end of the parent cell, and then this one eventually pops off, right, and then grows into a normal yeast size. And so this is an example of yeast. So nothing much to look at. They're just little round dots. And I'm actually looking for a budding cell, but I don't see it. Anyhow, oh yeah, here's one. There's one, right? There's a yeast that is budding. This is just a really great picture of spores at the end of the stalk of mold. So you can see how many there are, in this image. This is just another image of your mold. So we will be looking at something like this in the future. But again, this is the hypha, right? Those cells, those rectangular cells that grow and make a line. And then at the tip of the hypha, we have a specialized structure that makes our spores. And this one's called a sporangium, and we'll learn this later, but a sporangium is a kind of structure that makes spores. So this whole thing is not one spore. If you look inside how it has those little tiny dots, those would be the spores inside. And of course, the macro view, right? We have some moldy bread, and then the sort of view under like a dissecting scope or a magnifying lens, you could see that. Some of our fungi are dimorphic. Dimorphic means two, right? Di is two. Morphic is for the morphology or shape of the cell. So some pathogens can take on those two shapes depending on the temperature. So in the environment, which is usually at a cooler temperature, they form these hypha, and they'll form, these spore making structures, and the spores will be liberated into the air. If we were to inhale these spores and we had a dimorphic fungus, then they would form yeast cells. So because it's warmer. So at a warmer temperature, the yeast form takes hold. So this kind of dimorphism that depends on temperature is called thermo, right? We want to put the word thermo in front of dimorphic. So thermodimorphism, I won't go through the whole word, but thermodimorphism is this, this phenomenon where you have one fungus species having both forms depending on the temperature, and depending on the temperature, they'll choose one. And you do want to know that when we get infected with these kinds of organisms, it does make the yeast form in the body. All right, so how do we get fungal infections? We can inhale the spores. Again, tons of spores are in the atmosphere. We can get them through a cut, right? Because they're in the dirt as well. Examples of this, you don't have to remember the examples for the test, okay? This will come later, but there is one fungus that causes a very severe disease in some people, and it's inhaled, and that's called histoplasmosis. Then there's one that's pretty common among gardeners. It's called rose gardener's disease because roses have those thorns, which makes it easy to penetrate the skin, right? And so you could pick up those spores that way. Other routes of fungal infection is cutaneous, meaning that you just touch it with your skin. And this could be something like ringworm. It can be, it's basically they tell you not to walk barefoot at the gym or at a spa because you can pick up fungal infections, and it would be this form, cutaneous inoculation, by contact with infected fomites or infected people. So you can get, like wrestlers, for example, if somebody has a yeast infection or like a ringworm infection, for example, you can pass it on to another person because all they need to do is touch the organism. A fomite is a word that refers to any object, so any non-living article that can also transmit the pathogen. So fomites could be anything. It could be a toothbrush, it could be a door handle, it could be a sink handle. It's anything that's just non-living that's able sort of used to can pass on an infection. And then we have opportunistic invasion. The word opportunistic, right? We looked at this in a lab, I think, before. It means that it is usually found on our body as a part of our normal flora. So a great candidate or a great example of this is our Candida albicans. So Candida is in our vagina, in the mouth. It's on the skin. And once it moves out of its normal place, let's say you get a cut or something, it can cause infection. Or if you have an imbalance of bacteria versus the yeast in your mouth, or there's some kind of overgrowth that can happen. And that's the opportunistic. So it's usually not going to be an issue, right? It lives in our normal flora. It's part of our body, and we live most of our life free of any issues with this Candida albicans. But if you disrupt where it is, or if there's an imbalance in the flora, or if you get very, very sick, like if you're immunocompromised, the yeast have an opportunity to overgrow. So you give it the opportunity and it will overgrow. So that's where the yeast infections in the vagina or the mouth is called thrush. When you have an overgrowth of yeast in the mouth, that's where that comes from. So this is our respiratory example of our histoplasmosis. Just showing you, nothing to really memorize here, just that we have these molds within the environment. And a lot of times for this one, bird droppings, because they're very nutritive, will be very, very ripe and have a lot of histoplasma growing. And so you can inhale those spores and it does infect your lungs. So you can also see that in the environment, we see hypha in this image. And then in the person, right, we see more of the yeast form. This is that picture of Rose Gardner's disease, right? This is subcutaneous infection. This fungus, if you get it in your body, it tends to travel down a lymphatic vessel. So it follows a vessel, as you can see here, and it pops up these ulcers every so often as it grows down the vessel. Now, here are some examples of some dermatophytes, which is going to be our cutaneous route of infection. The word dermato refers to skin, right? So it's a dermatophyte. The one that is the most common that you'll learn is tinea, right? So actually just learn that now. Tinea is the genus for our dermatophytes. So when we say ringworm or, jock itch, that is a tinea infection. And so you can, these are contagious. This is the ones that you can pick up just by touching something, right? Your fomites or other people. So you can get this through personal articles, public facilities, swimming pools. You can get in contact with infected animals or another person, sexual contact, and also mother to child. Ringworm, right, is a tinea infection, and ringworm of the nail is more common in women who are artificial nails. And I ask why. So you might think that, oh, okay, if you have an artificial nail, like a nail on top, you have created a very warm, dark space, and within that little gap for if you did have tinea there, it could grow. So that's why those nail infections are more common with people who wear artificial nails. Sometimes tinea can cause really awful inflammatory responses. So you can see here tinea capitis. This is basically a dermatophyte infection on the head. You can see the inflammation there. And this is an image of our opportunistic candidate. This is Candida, and Candida albicans is the full term. You should know Candida albicans, and the infection it causes is called candidiasis. So in the vagina, it's called vulvovaginal candidiasis, and in the mouth, it's called thrush. And so you can see this child here, it's baby, has this white growth on the tongue and also on the gums and lips. This is thrush. And the reason why babies have thrush sometimes is because their normal flora of bacteria haven't really been established yet. And then the yeast can overgrow. So a key thing to remember here is that on our bodies, we have a mixture of our yeast cells and we have bacteria. And they are both sort of in balance, right? Not one is overgrowing over the other because that would cause infection. But if you have a lack of our normal flora and you do have that yeast, then the yeast is able to overgrow. In terms of their cell wall, there are two important things to remember about fungi. They have a cell wall made of chitin, which is a kind of polysaccharide. And they also have this cholesterol molecule called ergosterol, which is different than cholesterol. It's different than the cholesterol we have in our cells. Let me just point them out here. So ergosterol. Cholesterol is a component of membranes, right? So we have a lipid bilayer membrane here. We have our little purple molecules here, and that refers to ergosterol. So it's a component of their membrane, and this ergosterol is clinically important because it is a target of drugs. It is a selectively toxic target. Okay, so drugs often target the ergosterol because it's selectively toxic. What that means is that because our cells don't produce that form of cholesterol called ergosterol, it is different enough that we can make drugs that just attack the ergosterol and not the cholesterol that we have in our cells. And so we can just target and destroy those fungal cells instead of harming our cells. So the trick about all these eukaryotes, not just fungi, but eukaryotes in general, right? We also have eukaryotic cells. So their cells are fundamentally similar to our cells. They have the same electron transport chain. They, metabolize glucose the same way. There's so many similarities. So to find drugs that are just going to target something special about the eukaryote is more difficult. So you're going to have a lot of antibacterial drugs. Like there's so many antibiotics out there because bacteria have a prokaryotic cell and they're fundamentally different in many, many ways than our cells. So there's lots of targets to choose to kill them. But eukaryotes, not so much. So it is so ergosterol tends to be the one that a lot of antifungals will target. And due to the fact that we share the same cell as fungi, a lot of the drugs might have side effects because our cells might be harmed in the way of because of the medicine. So some of them can be life-threatening if they're not used. So, these are just some examples of our antifungals out there, but then don't memorize this. Okay. Treatment again, right? For dermatophytes specifically, right? So if it's growing on your skin, we can just target that with topical reagents or topical medicine. And so ringworm therapy, right? If you have a skin fungal infection, is basically like these guys feed on dead epidermal tissue, and you can block the multiplication of them and the fungus because our skin is constantly replacing itself. Like every month or so, our new skin cells move up to the top of our skin, they die, and then they fall off. So knowing that the fungus is growing on that top layer, it's sloughed off with the skin. So sometimes treatment is going to be, works by speeding up that sloughing off the skin, putting a topical antifungal on there. So if you have ever used the antifungal, sometimes it causes your skin to get really dry, and this is due to that sped up reproduction. Another interesting guy here, Aspergillus, is clinically significant. This is an organism that can grow on plants, and it forms a toxin called aflatoxin, and it can cause cancer. So any molecule or any sort of thing that can cause cancer is called a carcinogen. So aflatoxin is carcinogenic. So substances in tobacco smoke are carcinogenic as well, right? Because we know smoking causes lung cancer. And an interesting side is that, they can look for this because aflatoxin will grow in our UV light. All right, let's take a break.